There is a gland called the hypothalamus that exists in the brain and produces the hormone GnRH. This hormone causes the pituitary gland to secrete two hormones that control the growth and ovulation of the ovarian follicles. The levels of FSH increase during the menstrual cycle and cause the follicle to grow. The follicle continues to grow in size and produces increasing quantities of estradiol. Levels of progesterone remain low. In the uterus, the lining covering the inside of the uterine cavity, the endometrium, grows thicker. This is due to the action of estradiol. In the middle of the cycle, the LH surge results in ovulation. As a result of the rise in progesterone, following ovulation, the endometrium transforms itself in preparation for the reception of the embryo. At the level of the ovary, the follicle transforms into the corpus luteum, producing progesterone, which maintains the quality of the endometrium, facilitating implantation. If there is no implantation, the corpus luteum degenerates, with reduced production of progesterone, and the endometrium starts to shed the uterine cavity lining, resulting in bleeding, that is, menstruation. At the start of the menstrual cycle, a cohort of follicles starts to grow, but generally only one continues to develop. This follicle is referred to as the dominant follicle. In its interior, the follicle contains an immature oocyte. This follicle increases in size by 1 to 2 millimeters per day. The follicle produces estrogen and causes the lining of the uterus, the endometrium, to thicken. Around day 12 to 14 of the cycle, the follicle has grown to the size of about 21 millimeters. The rupture of the follicle ensues, which is called ovulation, and the mature ovule is released. The fallopian tube's fimbriae move towards the ovary and catch the oocyte that stays near the ampullary zone of the tube, waiting to be fertilized. If sexual intercourse occurs, the semen deposited in the vagina starts its ascension into the tube, and the sperm eventually arrive at the oocyte. One of these will succeed in penetrating into the interior of the oocyte. One day following fertilization, a cell with two nuclei is observed, at which point in time it is called an embryo, and it begins to divide itself. 2 days after fertilization, the embryo is made up of 2 to 4 cells and moves along the tube in the direction of the uterus. At the end of 4 days, the embryo continues to divide itself, at which point it is called the morula, and the interior of the embryo has an area filled with liquid, and this is called the blastocyst. The blastocyst migrates to the uterus and sheds the surrounding membrane, process of hatching, and the embryo begins to implant in the endometrium of the uterus. The function of the spermatozoid is to transport 23 chromosomes to the interior of the oocyte that contains another 23 chromosomes. It is divided into the head, with the nucleus containing 23 stored chromosomes. The nucleus is covered in its anterior 2/3 by a cap, the acrosome, which permits the perforation of the pellucid zone and penetration of the ovum with a drill-like motion. In the middle section, energy is produced that is used to move the tail. The sperm are propelled from the base of the vagina to the fallopian tube, where the oocyte will be fertilized. The male genital system is composed of 2 testicles that produce sperm, 2 seminal tubules that connect the testicles with the penis, 2 seminal vesicles, and a prostrate gland that produces seminal plasma. The seminal tubules are comprised of the epididymis and the vas deferens. The sperm are stored in the epididymis. The testicles consist of 8 to 12 seminiferous tubules that are connected by a straight tube with a rectular space called the rete testis. The rete testis connects the epididymus outside the testicle via the ductus deferens. The epididymis is very convoluted and, when stretched out, measures 6 meters. The sperm remain here for 20 days and mature, acquiring mobility. In the seminiferous tubal lumen, two types of cells are distinguished: steroli or nutritious cells and germinal cells. Between this, spermatogonium in contact with the tubal's walls are observed, spermatocytes, spermatids, and spermatozoids. The cells resulting of the meiotic division contains half of the chromosome. 46 chromosomes of the spermatogonium goes to 23 in the spermatozoid. The semen sample has to be obtained through masturbation after an abstinence of 3 to 5 days. The ejaculate is deposited in a collection pot. Once in the lab, the semen sample is prepared. Then, in order to select the sperm with the best motility, test tubes are used in which media of different densities are contained that allow for the separation of immotile semen, seminal plasma, cells of detritus that are also present in the ejaculate. An initial centrifugation is performed and the sperm are collected with a pipette. Further rounds of washing and centrifugation are done, after which the supernatant is discarded and the sediment is resuspended. The final sample obtained is evaluated under the microscope, and finally the sperm is stored in a test tube. The sperm that has been prepared is loaded into a syringe to which a catheter is attached. Then the speculum is introduced into the vagina, allowing the entrance of the neck of the uterus to be made visible. The catheter that has been charged with sperm is introduced high into the uterus, and the sperm ascends through the fallopian tubes, arriving at the area where the oocyte is located, and fertilization of the oocyte is facilitated. A needle is attached to the vaginal ultrasound probe. Both are introduced into the vagina. Under ultrasound guidance, the needle is inserted through the vaginal wall towards the ovary and is introduced into one of the follicles. The content of the follicle is aspired and is passed through a closed circuit of tubes to the test tube. This test tube will be analyzed by a biologist to separate the ovum from the follicular liquid. Once aspiration of the follicle is finished, the needle is inserted into the next follicle, which is then aspirated. This process is repeated until all the follicles have been aspirated. Once the oocytes have been obtained, they are left in a culture medium distributed in 4 wells. The surrounding cumulus cells are gently stripped off the oocyte. Parallel to this, semen obtained through masturbation is prepared, separating the mobile from the immobile sperms and then concentrating them. In intracytoplasmic insemination, one of the best sperm is selected. The tail is immobilized and is aspired into a fine needle. The oocyte is fixed with a micropipette and the other side is pinched by the needle and the sperm is injected into the oocyte. Once the oocytes have been obtained, they are left in a culture medium distributed in 4 wells. Parallel to this, semen obtained through masturbation is prepared, separating the mobile from the immobile sperm and then concentrating them. There are 2 types of insemination of oocytes: the conventional way and using ICSI. In the conventional way, a microdroplet of concentrated sperm is added to each of the wells containing the ova. In intracytoplasmic insemination, one of the best sperm is selected. The tail is immobilized and is aspired into a fine needle. The oocyte is fixed with a micropipette and the other side is pinched by the needle and the sperm is injected into the oocyte. Once the oocytes have been obtained, they are left in a culture medium distributed in 4 wells. Parallel to this, semen obtained through masturbation is prepared, separating the mobile from the immobile sperm and then concentrating them. In vitro fertilization, a microdroplet of concentrated sperm is added to each of the wells containing the oocytes. One day after fertilization, the embryos are observed under the microscope. If fertilization has occurred, a single cell is seen containing 2 nuclei, male and female. 2 days after puncture, the embryo divides itself and contains 2 cells, with 1 nucleus per cell, which in turn starts to divide into an embryo of 4 cells if its development is optimal. 3 days following puncture, the embryo starts to divide and consists of 8 cells if its development is optimal. On day 4, the embryo divides a number of times, becoming a morula. This is when it begins to become compact. On day 5, the embryo contains more than 100 cells. In the center, there is a fluid-filled cavity. Finally, the blastocyst will shed the membrane that surrounds it. This is called hatching. The embryos selected for transfer are loaded into a special embryo transfer catheter. The vagina is opened using a speculum. The embryo transfer catheter is introduced and, under ultrasound guidance, it is inserted until it is 1.5 centimeters from the base of the uterus. The embryos are introduced into the uterine cavity. The catheter is then carefully removed. Genetic pre-implantation diagnosis allows the detection of embryos that are affected by inherited single-gene diseases, such as those that are due to changes in the number of chromosomes. This is performed in embryos grown in vitro on day 3 of development. The diagnosis of single-gene disease is made using the technique of polymerase chain reaction, PCR. It is necessary to biopsy 1 or 2 of the embryonic cells, which need to be placed in test tubes containing a solution that will lyse the cell, liberating the genetic material for subsequent examination. After the amplification and sequencing of the gene region for analysis, it will be determined whether the embryos are affected by the disease. The gene diagnosis of numerical or structural anomalies of the chromosomes is performed using the technique of fluorescence in situ hybridization, FISH. It is necessary to biopsy an embryonic cell and fixate the nucleus on a slide. The nuclei are hybridized in the lab with special fluorescent probes to determine a few regions of the chromosome. In this manner, the healthy embryos can be differentiated from the affected ones. On day 6 after fertilization, the embryo, which is in the blastocyst stage, ruptures the enveloping membrane and subsequently sheds it. This is referred to as hatching. The embryo is ready for implantation into the endometrium. With assisted hatching, an embryo is clamped by a micropipette, and on the opposite side, the most outer layer called the pellucid zone, is breached using an acid tie rod. In this way, a kind of tunnel is created in the zone. The aim of this is to facilitate the exiting of the embryo out of its membrane to make its implantation more likely. The objective of hysterosalpingography is to check for the existence of anomalies in the uterine cavity and to ensure the permeability of the tubes. In order to do this, a speculum is inserted into the vagina until the neck of the uterus becomes visible. An applicator is attached to the neck of the uterus, where it is then fixed. Then a contrast agent is injected. In this manner, the contrast agent can be visualized as it passes through the uterus and the tubes, where it exits into the peritoneal cavity. In this hysterosalpingography, the accurate uterus and the left patent tube are visualized. The right tube is not patent. During the first 3 days, an ultrasound scan should generally be performed to rule out any pathology of the ovary and endometrium. If everything is normal, the patient starts to inject FSH subcutaneously. This hormone stimulates the growth of the follicles in the ovary. The dose applied is low since the objective is to have only 1 or 2 follicles growing. At the end of about 5 to 7 days of stimulation, another scan is performed to determine the number and size of the follicles, as well as the quality of the endometrium. On this day of stimulation, the follicle should reach a size of 13 to 15 millimeters. At the end of 2 or 3 days, a third scan is performed. When the follicle has reached a minimum size of 18 millimeters, ovulation can be triggered with the hormone HCG, which can also be injected subcutaneously. Ovulation occurs 30 to 40 hours after this injection. This is the reason that insemination must occur within this window of time. After insemination, a supplement of progesterone is administered vaginally for 2 weeks. A GnRH analog agonist is administered nasally or subcutaneously from day 21 of the menstrual cycle. Following an initial flare effect of LH, pituitary release of LH is suppressed, thus preventing premature ovulation of the follicles during stimulation. Around the time of menstruation, a scan is performed to check the thickening of the endometrium of the uterine cavity and rule out the presence of cysts in the ovaries. If everything is normal, subcutaneous daily injections of FSH is initiated. This hormone facilitates the growth of the follicles in the ovary. The dosage is established for each patient and is adjusted in consecutive checks. The objective is to have 4 to 7 follicles grow per ovary. At the end of approximately 4 to 6 days of stimulation, another scan is performed to check the number and size of the follicles, as well as the quality of the endometrium. Every 2 or 3 days a new scan is performed. When at least 3 follicles have grown to the size of at least 18 millimeters, final follicular maturation is triggered with the hormone HCG. The ova that are within the follicles will begin to mature. Oocyte recovery is undertaken 36 hours later. Following the procedure, a daily supplement of the hormone progesterone is given vaginally. 2 to 5 days after oocyte recovery, the embryos are transferred. 2 weeks later, a pregnancy test is performed. In the first days of menstruation, a scan is done to check the thickening of the endometrium of the uterine cavity and to rule out the presence of cysts in the ovaries. If everything is normal, the GnRH analog agonist is administered. 1 or 2 days later, subcutaneous daily applications of the hormone FSH is initiated. This hormone facilitates the growth of the follicles in the ovary. The objective is to have 4 to 7 follicles grow per ovary. At the end of approximately 4 to 6 days of stimulation, another scan is performed to check the number and size of the follicles, as well as the endometrial thickness. Every 2 or 3 days a new ultrasound scan is performed. When at least 3 follicles have grown to the size of at least 18 millimeters, final follicular maturation is triggered with the hormone HCG. Oocyte recovery is undertaken 36 hours later. Following the procedure, daily administration of the hormone progesterone is given vaginally. 2 to 5 days after oocyte recovery, the embryos are transferred. 2 weeks later, a pregnancy test is performed. In the first days of menstruation, a scan is done to check the thickening of the endometrium of the uterine cavity and to rule out the presence of cysts in the ovaries. If everything is satisfactory, subcutaneous daily injections of FSH is initiated. The dosage is established for each patient and is adjusted in consecutive checks. The objective is to have 4 to 7 follicles growing per ovary. At the end of approximately 4 to 6 days of stimulation, another scan is performed to check the number and size of the follicles, as well as the quality of the endometrium. After 6 days of stimulation, a GnRH antagonist is injected subcutaneously. This prevents premature ovulation. Every 2 or 3 days, a further scan is performed. When at least 3 follicles have grown to a size of at least 18 millimeters, final follicular maturation is triggered with the hormone HCG. The oocytes that are within the follicles will begin to mature. Oocyte recovery is undertaken 36 hours later. Following the procedure, a daily supplement to the hormone progesterone is given vaginally. 2 to 5 days after oocyte recovery, the embryos are transferred. 2 weeks later, a pregnancy test is performed. In the first days of menstruation, ultrasound scan is performed in which the thickness of the endometrium is determined and the presence of cysts in the ovaries is ruled out. If everything is normal, the administration of estrogen by the oral route or by means of an estrogen patch is initiated. The addition of a GnRH analog nasally or by subcutaneous administration is optional. The dosage of estrogen is increased in a progressive fashion. Approximately 7 days after the start of stimulation, a new scan check is done to determine the quality of the endometrium. The scans are repeated until the endometrium has reached a certain optimum size and appearance. When this occurs, daily progesterone is administered, and if a GnRH agonist was used, this is also stopped. When progesterone has been administered for at least 3 days, the transfer of embryos that have previously been thawed can take place. After the transfer, progesterone and estrogen are continued to be administered. 13 days after the transfer, the pregnancy test is performed.